Hi, Assalamualaikum and very good day to the examiner. My name is Aziemah. I am final year student of Bachelor Degree of Science Biology. Today, I would like to present about my review paper on diet quality impact on growth rate of freshwater rotifer Brachynes caliciferus by using nanocorrosive species and laminaria species as food. My supervisor is Prof. Madya Abdul Razak bin Abdul Rahman. Next, for the introductions of this review paper, which is the name of rotifera come from two Latin words which is rota, wheel and ferens. To build at its same name means like wheel barriers. This specifies that the ground or the silla in the vicinity the mouth of rotifers. Rotifera are the one of the smallest metazoan of which more than 1,000 species has been described, 19% which is live in freshwater habitat. One of the common genera is Brachynes. Besides, it is recognized that feed is an important component of the practice of aquaculture progressions. Rotifer is very important in aquaculture as it is vital food for noble larvae of many fish and crustacean species. Besides, in rotifer Brachynes caliciferus has specific nutritional requirements that must be fulfilled in order to produce healthy cultures. There are several problems today in producing high productions of rotifer B. caliciferus, which typically declines in growth rate and cannot live long. From this problem, the growth rate of the B. caliciferus will be affected, which can easily to die. This problem can make the growth rates of the rotifer decrease, and these conditions affect the larvae of the fish that they eat and can cause the productions lowering and decrease. Nutrient loss can reduce the nutritional values of rotifer for larvae and make it difficult to conclude on the intake of fish larvae nutrients. However, although rotifers are tolerant of many abiotic influence, the mass culture collapsed suddenly and hatcheries seek to fix this problem by growing more rotifer. So, there are few challenges they have to face, which are to control the effects on the rotifer by different type of foods, the stability of foods, and the concentrations of foods that the effect of the cultural condition to grow in aquaculture. Therefore, the purpose or the objective of this study is to determine the growth rate of the rotifer B. caliciferus, where they can grow in different nutrients of foods. Besides, this study also to determine the stability of the rotifer B. caliciferus with different type of food given. Not only that, this research also to study the conditions of the cultures of the rotifer B. caliciferus given by different concentrations of food. So, the significance of study of this review paper is the growth rate of the rotifer is very significant to identify the effectiveness of seaweed laminarius sp, for example, cap seaweed, to increase the rate growth of the rotifers. Seaweed is a macroalgae that cheapest, easier to treat, suitable food that is always available and easy to find. Producing huge amount of rotifer is very important to feed the larvae of the fish, where the rotifer is rich in the amino acids that become a suitable live food for fish, where it's able to increase the survival of larvae. This study also can help people to know better about the effect of the food that contains nutrients that help rotifer to grow. Besides, the finding will benefit to society who involved in agriculture. In addition, the Brachynes caliciferus is the platonic rotifer that found in fresh water. Most of rotifer B. caliciferus has approximately the same body length in about 220 to 280 micrometer and body width 124 to 300 micrometer. Even though B. caliciferus is planktonic rotifer, ecophysiological trait considerably depends on the species body size. With optimum pH of 6 to 8, Brachynes caliciferus tolerate temperatures between 15 Celsius to 31 Celsius. To the next slide, which is morphology of rotifer B. caliciferus. B. caliciferus rotifer is small metazoan, approximately 200 micrometer metazoan, which is a phylum rotifera, are popular and sometimes very evident in nutrient-rich freshwater in zooplankton ecosystem. B. caliciferus are bilateral, which means that body of this rotifer is bound by symmetrical bilaterals. Furthermore, the body consists of 4 regions, which is head of corona, neck, body, and foot. According to Gilbert 2018, the ciliated corona is used for locomotion and for producing feed flow from which tiny, mostly 2 to 20 micrometer particles. Next, for the reproductions of Brachynes caliciferus, Brachynes caliciferus reproduce by asexual and sexual reproductions. In the subclass Monogonata, life cycles haplodiploid with cyclic pathogenesis, turbinophase amniotic females, while sexual reproduction mexes contain mictic diploid females producing haploid eggs, which is, if fertilized, it produces diploid embryo or cysts in the old literature are called resting age.
There are few factors that affect the growth rate of bee caliciferous. The first is effect of different types of nutrient foods. Based on the previous researchers, Lucas and Socket 2019 stated that rotifers are hardly and easily to cultivate on a large range of foods. According to Trish and Darby 2000, they stated that highest bee production occur when rotifers feed by Isocyanus galbana, while Wacker and Christberg 2012 stated that bee caliciferous restricted more than one nutrient like steroid and other amino acids. Next is different concentrations of foods. The concentration of food affect the growth of rotifer. This is proven by Savaja 2019 that population of bee caliciferous influenced by food quantity. For example, food density 10 to 20 times 10 power of 6 per ml nanochlorosis was suggested for culture bee satilis, but for optimal food density is 0.1 times 10 power of 6 per ml. In my previous studies, nanochlorosis is the best fit on grow weight rotifer compared to seaweed because nanochlorosis species is small alga which is 2 micrometer with a very high content of lipids, protein and calories. Lastly, stability of rotifer based on different types of food. In my previous study stated that rotifer are opportunists that react very quickly to any change of food types. For example, dietary medium that contain albumin or in combination with this will not help the growth of the rotifer. Next, for the research finding in nutrient source of laminary SP and nanochlorosis SP in source of protein as mentioned by Beckley and Hayes 2017, seaweed and microalgae are considered and regarded as viable source of protein. This study related to protein key factor in determining the potential health benefit of a food product since it is potent nutrient for growth. For brown algae is characterized by the presence of the pigment of fucoxantins, and the reason why choosing seaweed because based on the review had been done, brown algae are unique among the algae as they can only be found in multicellular form, but at the same time, brown algae had the lowest protein content compared to red and green algae. Next, for the green algae, like uva found in the brackish water, these species are very attractive to research because they are packed with mineral, protein, and vitamins. For the red algae, it have a high level of protein compared to green and brown algae. And the lastly is nanochlorosis SP. Another study conducted showed that microalgae are high in 3-HUFA such as nanochlorosis SP, and it was considered very good for growing rotifer.
On this review paper, bird culture method is most suitable for cultures rotifer bee caliciferous. Bird culture method used to produce mass productions of the bee caliciferous. It is possible to culture the rotifer until it produces a large population density. According to Yoshimatsu and Hussein 2014, the bed culture system is the first method to culture bee satilis. The bed culture method was used to produce the mass production of bee satilis was to be used during the experiment. As a conclusion of my review papers, overall rotifer or species Brachynes caliciferus has been described as an important food source and have a variety of characteristics that make them desirable as live food in aquaculture. There are several factors that affect the growth rate of the rotifer, such as effect of different types of foods. Different types of food have different types of nutrients in the foods. Based on research that have been done, the population of rotifer increased dramatically when cholesterol supplementations and amino acid supplementations in the foods, and indicates that a dietary source of steroids needed to achieve high population growth rate of rotifer. In addition, food concentrations also part of factors that affect the growth rate of the rotifers. The stability of the rotifer depends on the quality of the nutrient food supplies. The results stated that microalgae, which is nanochlorosis sp, are widely used in the rotifer diet than macroalgae like laminaris sp, brown algae. Moreover, nanochlorosis sp are high in 3-HUFAs, and it was considered very good for growing rotifer compared to laminaris sp. Therefore, in order to increase knowledge in the study of bee caliciferous species, in addition to study about the freshwater rotifer species with different growth rates, effect could provide recent and up-to-date actualities cultures. From me, thank you for your time for listening my presentations review papers. Have a good day.